Bismillahirrahmanirrahim. Assalamu alaikum. In this video, we are going to talk about pyruvate oxidation or link reaction. Pyruvate oxidation is actually the conversion of pyruvate into acetyl coenzyme-A by an enzyme complex known as pyruvate dehydrogenase complex. In the previous video, we discussed glycolysis, which results in the breakdown of glucose into pyruvate molecules. These pyruvate molecules enter mitochondria and is converted into acetyl coenzyme A before it enters the next step of respiration, that is citric acid cycle. The process is known as pyruvate oxidation, pyruvate decarboxylation, or oxidative decarboxylation of pyruvate molecule. It is a combination of oxidation and decarboxylation. The reaction is also known as oxidative decarboxylation. The process is also known as link reaction because it connects or links glycolysis and Krebs cycle. Pyruvate molecules produced in glycolysis does not enter the Krebs cycle directly. It enters mitochondria and is oxidized into a 2-carbon molecule that is known as acetyl group. The pyruvate oxidation completes in 3 steps, that is decarboxylation, oxidation, and conjugation. Decarboxylation is the removal of carbon in the form of carbon dioxide. Carbon is removed from pyruvate molecule in the form of carbon dioxide, and the process is known as decarboxylation. Oxidation is actually the removal of hydrogen atom from pyruvate and transfer the hydrogen atoms to NAD, which is reduced to NADH. And finally, the acetyl group unites with a coenzyme-A to form acetyl-CoA. And this process is known as conjugation. So these are the 3 steps of a complex reaction which is catalyzed by the enzyme pyruvate dehydrogenase complex. The products of pyruvate oxidation are reduced coenzyme NADH, carbon dioxide, and acetyl coenzyme A. In acetyl-CoA, a thioester bond links acetyl group to a sulfur-containing coenzyme-A. The coenzyme-A consists of a dinucleotide and one of the B vitamins. The coenzyme-A part of acetyl-CoA, it acts as a transporter for the transfer of acetyl group. The main function of coenzyme is to deliver the acetyl group to mitochondria, where it enters the citric acid cycle or Krebs cycle. This is the diagrammatic representation of pyruvate oxidation. This is pyruvate molecule produced as a result of glycolysis. This molecule, this pyruvate, enters the mitochondrial matrix through a transport protein. This is the transport protein. So the pyruvate molecule enters the mitochondrial matrix through this transport protein. After entering the mitochondrial matrix, the first step is decarboxylation, that is the removal of carbon dioxide. This part of pyruvate molecule is removed in the form of carbon dioxide, and this is known as decarboxylation. After decarboxylation, the remaining part, the acetyl group, is oxidized by removal of hydrogen atom, and in the process NAD is reduced into NADH. So these 2 reactions are collectively known as oxidative decarboxylation. And the third step of the reaction is a combination of coenzyme A with the acetyl group, which results in the formation of acetyl-CoA. So this is the end product of pyruvate oxidation. This acetyl coenzyme A enters the Krebs cycle and reacts with the oxaloacetic acid, which is the first reaction of citric acid cycle or Krebs cycle. And that will be discussed in the next video. So this was all about pyruvate oxidation or link reaction. Thank you for watching, and please subscribe the channel for upcoming videos.